Hello. So I welcome you all in my today's lecture. So today it will be our summary lecture, which I am going to present right now. So just let me share the entire screen first. So today it is the summary lecture for the NPTEL course of the medicinal chemistry, and we have till now completed our 12-week course, and we have completed the 12 lectures. 12 all the 12 lectures were based on your 12-week course content. So we have completed the 12 lecture with the 12-week course content, and today it is our summary lecture. So what we will do in this summary lecture is I will summarize the complete session today, and I will point out few things which I have not discussed in detail. And also I will first what I will do is I will just give the some MCQ questions. I will show you particular question, wait for some time. After that I will show you the correct answer for that question, and we will discuss it a little bit. Followed by I will summarize the entire course. I have selected a few topics which I am going to show you today, and I am going to discuss the topics with you in detail. Few topics I have already discussed and few things I haven't discussed earlier. So I think it is a good option for us to have such a summary class where I can summarize the complete lecture. So let us start with today's session. So very good evening to all. Just let me introduce myself. So I'm Mousumi Deb. I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So the first question of today's session, I will let me show you. So the first question of today's session, it says that which of the following drug is withdrawn from the market due to its damaging effect on the liver cells. Your options are: number one is Practolol, number B is Tylenic acid, number C is Serivastatin, and number D is Phenylbutazone. So which of the following drugs is withdrawn from the market due to its damaging effect on the liver cells? I will wait for a minute, and then I will show the correct answer for the particular question, and then I am going to discuss it. So this particular question is related to the post-market surveillance of the drug or of the clinical trial. So we have already discussed that how clinical trial happens. So what are the criteria of the clinical trial? When the drug is approved for the market, after the preclinical trial, when the drug is tested on the humans in the clinical trials at I mean small number of group of the people. After that, the drug is released in the market, and after some time the phase 4 clinical trial actually happens, and where it is checked that whether the drug is generating any kind of side effects after its after its introduction to the market, when it is applied to a bigger population of the patients. So that is tested in the post market surveillance. Okay, so this question was related to that particular topic. So let me show you the correct answer. The correct answer will be number B, tylenic acid. Tylenic acid is a drug which is withdrawn from the market due to its damaging effect on the liver cell. So this is the structure of the tylenic acid. So what is the phase 4 clinical studies? As I have mentioned to you earlier, so the drug is now placed in the market and it can be prescribed, but still it monitors for the effectiveness and for any rare or unexpected side effects. In a sense, this phase is never ending process, as unexpected side effects may crop up many years after the introduction of the drug. In the UK, the Medicines Committee runs a voluntary yellow card scheme where the doctors and pharmacists report the suspected adverse reactions of the drug. This system has revealed serious side effects for a number of drugs after they have been put in the market. For example, the beta blocker Practolol had been withdrawn from the for several years because some patients suffered blindness even the death. So beta blocker, I think I have introduced this beta blocker. What is a beta blocker? What kind of therapeutic it is? It is an antihypertensive drug. We have already discussed it in our earlier lectures. So the toxic effects were unpredictable and are still not understood. So it has not been possible to develop a test for this effect. Diuretic agent tyrannic acid. Diuretic agent also. So tyrannic acid is a diuretic agent. Diuretic agent is also a hypertensive agent, antihypertensive agent, means which is used to lower down the blood sugar. Sorry, it's used to lower down the blood pressure in the patients. Okay, so it had to be withdrawn from the market because it damaged the liver cells 1 out of every 10,000 patients, which is a quite a similar I mean quite a large number, because if you think there are so million people in the world, so 1 out of 10,000 is quite a high number. So the anti-inflammatory agent phenylbutazone can cause a rare but fatal side effect in 22 patients out of every million treated with the drug. Such a toxic side effect would clearly not be detected during the phase 3 clinical trial. Because, so why this kind of side effects are not detected during the phase 3 or phase 2 or any other phase clinical trials? Because all the clinical trials which are earlier, so these are performed on a smaller number of people, which I have already mentioned you, right? And only this particular phase 4 clinical trial is done on the large number of people. That is why some side effects are not detectable in the phase 3 or any other phase clinical trial, but they can be detectable during the post market surveillance, which is a post phase 4 clinical trial. More recent example is the serivastatin, which was marketed as a potent anti-cholesterol drug. Unfortunately, it has to be withdrawn in 2001 as a result of the adverse drug-drug interaction, which resulted in the muscle damage and the death of the 40 people worldwide. Okay, so the correct answer of this question was a tyrannic acid. Now I will go to the next problem. So problem number 2 says, which of the following statements is true regarding the two-dimensional electrophoresis? The options are: number A, proteins are separated on the basis of the charge; number B, proteins are separated on the basis of the molecular weight; number C, the proteins are separated on the basis of pi, and or which is the isoelectric point; number D, proteins are separated on the basis of both molecular weight and pi. So which of the following statements is true regarding the two-dimensional electrophoresis? Again, I will wait for 1 minute and then I will show the correct answer. So the statement, which of the following statement is true about the two-dimensional electrophoresis? So the 2D electrophoresis and the electrophoresis are the one of the techniques by which the proteins can be separated. There are one-dimensional electrophoresis also, along with the two-dimensional electrophoresis, and there are several other techniques which are used to separate the proteins based on their either molecular weight or isoelectric pointing or any other their physical their property. Okay, so this question is related to the 2D electrophoresis. Now let us see what will be the correct answer for this question. So the correct answer of this question will be number D, proteins are separated on the basis of both molecular weight and pi. So in 2D electrophoresis, the proteins are separated on the basis of both molecular weight and its isoelectric point. So what is the isoelectric point? So basically we all know amino acid has a basic amino side and a acidic cerebelloid group. Okay, so the both having this both acidic cerebelloid group and the basic amine group makes the protein charged molecule. Like in every page the protein has a certain charge, because and the at some point at some page the protein has a zero charge. It is neutral because it has an equal number of positive and negative functional group, and the overall charge of the particular amino acid becomes zero, which is known as this isolated that particular page. When the particular amino acid has a I mean the overall charge zero, it is known as the isoelectric point or its pi. Okay, so That is so let us now understand what is the principle of 2D electrophoresis. So in 2D electrophoresis, the proteins are separated as per their isoelectric point and protein mass. Separation of the proteins by the isoelectric point is known as the isoelectric focusing, or IEF. When a gradient of pH is applied to a gel and an electric potential is applied across the gel, making one or end more positive than other. At all pH values other than their isoelectric point, proteins will be charged. If they are positively charged, they will be pulled toward the negative end of the gel, and if they are negatively charged, they will be pulled to the positive end of the gel. The proteins applied the first dimension will move along the gel and will accumulate at their isoelectric point. That is the point at which the overall charges of the protein is 0. Okay, so let me continue it. So as I have mentioned earlier, that in the isoelectric point, or PI, is when the overall charge of the protein is 0 as a neutral charge. In the separating proteins by one mass, the gel is trapped with the sodium dodecyl sulfate, SDS. So SDS page electrophoresis is another technique which can also be applied for the separation of the proteins. Okay, so what is the principle? In this case, the gel trapped with the sodium dodecyl sulfate, or SDS, along with other reagents. So this SDS denatures the protein, that is unfolds them into long and straight molecules, and binds a number of SDS molecules roughly proportional to the protein's length. Because a protein's length when unfolded is roughly proportional to its mass, since the SDS molecules are negatively charged, as a result that all the proteins will have approximately the same mass to charge ratio as each other. So it is overview of the 2D gel electrophoresis. So first one is the isoelectric focusing, is the first dimension. So this is the how the decreasing pi, so this is the decreasing pi of the proteins. Okay, and here that's how, as you can see, the bands appear and after that isoelectric focusing, the gel is placed on the polyacrylamide SDS polyacrylamide acrylamide gel, which is also known as SDS page, and it is keeping like on the horizontal. Okay, so first it is vertical, then horizontal, and then second dimensional electrophoresis happens. So the particular proteins are separated like this. So this is the 2D, the two dimension. In the y-axis, it is the decreasing molecular weight, and on the x-axis, it is the from left to right, it is the decreasing pi, and on the y-axis from up to down, it is the decreasing molecular weight. So this is how, that is the main principle of the 2D electrophoresis, and that is how, using 2D electrophoresis, that particular separation of the proteins happen. Okay, so this was the question related to how the separation of the proteins happen. And now I will go to the our next problem. So let me go to the problem number 3. So the problem number 3 says smallpox and the yellow fever are serious viral infections, but these can be tackled by which of the following? Your options are: number A, antiviral drugs; number B, natural product; number C, vaccination; number D, immunotherapy. Smallpox and yellow fever are serious viral infections, but these can be tackled by antiviral drugs, natural product, vaccination, or immunotherapy. So smallpox and yellow fever are serial viral infections, but these can be tackled by number A, antiviral drugs; number B, natural product; number C, vaccination; and number D, immunotherapy. Which will be correct? So basically, this question is related to the antiviral drug discovery. We have discussed a lot about various antiviral agents, how the antiviral agents work. We have discussed about it. Okay, so various antiviral treatment. There is a there are like immuno. So all of these are the antiviral therapies, like antiviral drugs, and vaccination, immunotherapy, and all these are the various method by which the antiviral therapy can. I mean, it can we can fight the viral infections by all these techniques. So now let us just check what will be the correct answer, how the particular yellow fever and the smallpox can be tackled. So the viral infection, the serious viral infections, smallpox and yellow fever can be tackled by number C, vaccination. So what is vaccination? So one of the ways in which we can protect ourselves from the viral disease is to use the vaccination, and a number of diseases that we looked at such as polio, measles and mumps are actually mainly targeted by the vaccinations. So smallpox and yellow fever are also sort of serious diseases which are encountered by the vaccination. So vaccination works by introducing the body to foreign material which bears a molecular similar into some component of the virus but which lacks infections nature of then the toxic effects. The body then has to opportunity to recognize the molecular fingerprint of the virus, that is a specific antigens, and the immune system is primed to attack the virus should it infect the body. The vaccination, usually a killed or weakened version of the virus is administered, so it does not lead to infection itself. Alternatively, fragments of the virus, the subunit vaccines can be also used if they display a characteristic antigen. Vaccination is a preventive approach and it is not usually effective on the patients who have already become infected. So this is how various techniques of the vaccination can actually happen. So this is the structure of the virus in the antiviral in the lecture when I was discussing the antiviral drugs. I have discussed how the viral infection happens, and also I have discussed the structure of the virus, what are the different components. So let me just do it one more time. So this is the envelope protein is present on the outer side of the virus, and okay, and this is the membrane protein also is there, and also we have the spike protein or the glycoprotein. Okay, and then we have the genomic material, the single strand RNA and plasmid, and also so there are so various enzymes like protease enzyme, integrase enzyme, and so on, which actually works inside of the virus. So this is a viral particle. So what are the different types of vaccine? So first is the inactivated vaccine based on the chemically and or physically inactivated virus grown in the cell culture. Live attenuated vaccine, this utilizes a weakened version with a limited replication extent of the living virus. Then we have the viral vector vaccine, adapts the existing successful and safe modified viral vectors to express the coronavirus protein. It is an example. Okay, and then DNA vaccine contains circular DNA that encodes the protein of interest. Then RNA vaccine uses mRNA packed in the lipid nanoparticle like this. Okay, this is an lipid nanoparticle. In it we have the mRNA, and also recombinant subunit vaccine composed of the synthetic nanoparticle coated with purified viral antigen like this. And virus like particles vaccine contains the multi-protein structure that mimic the virus yet devoid of any viral genome. So these are the different types of the vaccines, or different types how vaccination happens. Vaccination has been a very good very important and very active way to protect the cells from the future viral infections. Okay, so this was our third question. Now let me go to our next slide with our problem number 4. Okay, so problem number 4 says which of the following is a nitrosourea alkylating agent? Your options are number A lomustine, number B carmustine, number C streptozotocin, and number D all of the above. Which of the following is a nitrosourea alkylating agents? Lomustine, carmustine, streptozotocin, or all of the above? Again, I will wait for a minute and then I will show you the correct answer and we will discuss it a little bit. Alkylating agents, I have already discussed it earlier with you. Say what are the alkylating agents, what are the what are the ways of the what are the clinical aspects of different alkylating agents, also along with their mechanism. I already have discussed it a couple of times. So it is a very important thing, the alkylating agent, and their mechanism of action is also. So there are several types of alkylating agents. Nitrosourea is one of them, and the question is regarding the which of the following is a nitrosourea type of alkylating agent: lomustine, carmustine, streptozotocin, or all of the above. 1 minute I will wait, and then I will show you the correct answer and we will discuss it in detail. So as I have mentioned you that alkylating agent has various clinical aspects, and one of them is they are very good anti-cancer agents actually. Okay, so now let us see which of the following will be the correct answer, which of the following will be the anti-cancer, no, which of the following will be the nitrosourea alkylating agent. Now let me show you. Okay, so so in the question number, okay, one second. Yeah. So the following, which is the not, which is a nitrosourea alkylating agent, the correct answer will be number D, all of the above. So lomustine, carmustine, streptozotocin, all of the above are the nitrosourea alkylating agents. So let us see the clinical aspects of this. So nitrosourea, the anti-cancer agents lomustine and carmustine were discovered in 1960s and are ethyl nitrosoureas which decompose spontaneously in the body to form two active compounds, an alkylating agent and a carbomylating agent. The organic isocyanide that was formed carbomylates glycine residues in the protein may inactive the DNA repair of the repair enzymes. The alkylating agent reacts initially with a guanine moiety on one strand of DNA and then with a guanine and cytosine unit on the other strand to produce the intrastrand cross-linking. Streptozotocin is naturally occurring nitrosourea isolated from the Streptomyces erythromonogenes. Okay, so this is the structure of the lomustine, this is the structure of the carmustine, and this is the streptozotocin, which has almost similar structure of the other one, but it has a little different structure because it is a natural product. Okay, so basically let us see what is the mechanism. So as you can see, this is the mechanism of action of nitrosourea alkylating agents. So we have two parts actually. First is the isocyanate part. So first is there is a hydroxyl group. It attacks on the this, it abstracts this proton, and this type of resonance happen, and we have this isocyanate which and carbomylation happens with the protein lysine residue, and it creates this kind of moiety. And other is this azo type of compound which is after releasing the nitrogen and hydroxide give rise to this alkylating agent. This alkylating agent give rise to the DNA alkylation with the DNA chain. Okay, and it causes the intra strand cross linking. So the nitrogen bases which are responsible for this type of cross linking is the guanine and the cytosine. This is the structure of guanine, this is the structure of cytosine. Guanine and cytosine, they take part in the hydrogen bonding with the three hydrogen bonding. So as you can see, if you look at carefully, here we have a carbonyl carbon. Okay, this carbonyl oxygen acts as a hydrogen bond acceptor, and this amine acts as a hydrogen bond donor. So this binds with this. This is the hydrogen bond donor, this is a hydrogen bond acceptor, this binds with this, and this is a hydrogen bond acceptor, this is the donor, so this bind with this. So that's how these three portion we have guanine and cytosine bind with each other and have the three hydrogen bond which we have learned so far. And alkylation by the nitrosourea happens on this acceptor position and also on this acceptor position on the cytosine, and this active acceptor position on the guanine. So these are the alkylation site on the guanine and cytosine for the nitrosoureas. So this was about the nitrosourea, and other alkylating agents I have already discussed you a lot about. So one very famous alkylating agent is phosphoramide, which I have or the the mustard, phosphate mustard. Okay, that I have discussed earlier also. So this one is related to the your nitrosourea alkylating agent, the different example, and the their different mechanism of action. Okay, that is I have discussed it. Now let us go to our next question, the problem number 5. So problem number 5 says which of the following is not a fourth generation cephalosporin. Your option are cephalexin A, cefoxitin B, ceftazidimine C, and cefpirom, which is D. So your options are number A and B, number B, A, B, and D, number C, A, B, and C, and option number D is C and D. So which of the following is not a fourth generation cephalosporin? Cephalexin A, cefoxitin B, ceftazidime C, and cefpirom D. I will wait for 1 minute, then I will show you the correct answer. Which of the following is not a fourth generation cephalosporin? So cephalosporin is very important antibacterial therapeutic along with penicillin. Okay, the cephalosporins are also a very large number of antibiotic group, but I earlier when I was discussing about it, I didn't given I think much I mean I didn't discuss about cephalosporin to a much extent. Rather, I discussed about penicillin more. So let us now understand what are the clinical aspects of cephalosporin, because these are also very crucial antibacterial agents. Again, I will wait for a minute, and then I will show the correct answer and will discuss about the cephalosporins. So basically, in case of the antibacterial agent, along with the penicillin, cephalosporins also hold a very good, place in the as a antibacterial agents. And among the cephalosporin, when you are reading it out, you have to remember what are the clinical aspects of the cephalosporin, and what are the examples of different generation of cephalosporin along their clinical aspect, and also the SAR of cephalosporin along with the SAR of penicillin is very important. You will find it in the book which I have mentioned you earlier, or you can just go through the lecture slides also. There is mentions that what is the SAR of cephalosporin, that is equally important as the SAR of the penicillin. Okay. So now let me show you which of the following will be the correct answer. So the following which is not a fourth generation cephalosporin, it is the correct answer is A, B, and C. So cephalexin, cefoxitin, and ceftazidime. These A, B, and C are not fourth generation cephalosporin. Only cefpirom is the fourth generation of the cephalosporin. Okay. This is the structure of the cefpirom. So this is the basic structure of the cephalosporin. Just like penicillin, it has also has a beta lactam ring, along with it also has a fused bicyclic system, and the stereochemistry of these hydrogens and these groups are very equally important, just similar to the your penicillins. Okay, so the clinical aspects of cephalosporin. So in general, cephalosporins are useful broad spectrum antibacterial agents for the treatment of the septicemia, pneumonia, meningitis, binary tract infections, peritonitis, and urinary tract infections. Cephalosporin C itself has been used in the treatment of the urinary tract infections, as it is found to concentrate in the urine and survive the body's hydrolytic enzyme. Okay, the first generation cephalosporin have good activity against the gram-positive cocci, and they can be used to treat some community-derived gram-negative infections, that is the infections that are not caught in hospital. Okay, they can be also used against the S. aureus and streptococcal infections when the penicillins have been avoided. Cephalothin is more active than the penicillin G against some gram-negative bacteria and is less likely to cause the allergic reactions. It can be also used against the beta-lactamase producing the S. aureus strains. Cephalexin is useful for the treatment of the urinary tract infections which do not respond to other drugs or which occur in the pregnancy. It is also useful in treating the infections of the respiratory tract, ear, skin and mouth. Cefaxolidine is recommended for the use as the prophylactic to prevent the infection when surgical procedures are used to implant the foreign bodies. Okay, second generation cephalosporin. So the cephalexin is the first generation along with these two. So second generation cephalosporin, in general, second generation cephalosporin have the variable activity against the gram positive cocci but increased activity against gram negative bacteria. Cefoxitin is active against the bowel flora, including the bacterium Dias fragilis and was once recommended for the peritonitis. Cefoxim has a wide spectrum of activity and is useful against organisms which have become resistant to penicillin. However, it is not active against the difficult bacteria such as P. aeruginosa. It is all used clinically against the Neisseria gonorrhoeae and also respiratory infections caused by H. influenza. Moraxilla catarrhalis and the susceptible strains of S. pneumonia is also used for surgical prophylaxis as well as the treatment of the Lyme disease. As a cefoxitism is used in the surgical prophylaxis and for the treatment of gonorrhea, meningitis and infections caused by the Haemophilus epilotis. Third generation cephalosporins have good activity against the gram-negative bacteria but vary in their activity against the gram-positive cocci. Their ability to attack the P. aeruginosa also varies from the structure to structure and they lack the activity against the MRSA organisms and Enterobacterium species. Ceftazidime is an injectable cephalosporin which has excellent activity against the P. aeruginosa as well as other gram-negative bacteria because the drug can cross the blood-brain barrier. It can be used to treat the meningitis. This is very important. Okay, because not many of the drugs they can cross the blood-brain barrier compared to other aminothiazole structure. Cefoxitism has a good activity against the streptococci but loses its activity against the strains of the methicillin susceptible Asorius, thus because the decreased binding affinity for the transpeptidase enzyme present in the Asorius. Ceftriaxone is used for surgical prophylaxis and as a prophylactic for the meningococcal meningitis. Fourth and fifth generation cephalosporin. The fourth generation cephalosporin have the activity against the gram-positive cocci and a broad area of gram-negative bacteria, including the P. aeruginosa and many of the Enterobacteria species. Ceftriaxone is administered as an intravenous injection or infusion and has been used against a variety of sensitive gram-positive and gram-negative bacteria. Ceftaroline fosamil has been licensed for the treatment of bacterial pneumonia and acute bacterial skin infections. So these are the clinical aspects of the cephalosporin. Sometimes it may feel like these are too much to remember, but what I can say you that these are the very important things, and if even if you cannot remember everything, I will suggest you to just take few examples of cephalosporin and try to remember their clinical aspects and along with their generation, which will be helpful because some cephalosporin are very unique and they are used in a very unique disease. So that's thing you need to remember. Okay, that is why I have given that clinical aspect portion, because I think you have to understand these things and also you have to remember these things. Okay, so now let us go to the next problem. So problem number 6 is augmentin is a life-saving drug and it is a mixture of number A oxacillin and ampicillin, number B amoxicillin and clavulanic acid, number C oxacillin and clavulanic acid, number D amoxicillin and pyruvic acid. So augmentin, a life-saving drug, is a mixture of which of the following? Oxacillin, ampicillin, or amoxicillin, clavulanic acid, or oxacillin or clavulanic acid, or amoxicillin and pyruvic acid. Again, I will wait for a minute. Then I will show you the correct answer and I will discuss it. So augmentin is a life-saving drug and it is a mixture of oxacillin, ampicillin, amoxicillin or clavulanic acid, oxacillin or clavulanic acid, or amoxicillin and pyruvic acid. So sometimes what happen, like we give a particular drug for a particular therapeutic aspect, but there are some components in our body, or there are some enzymes and something that harm that particular therapeutic agent, and that is why we have to give a second drug to just protect our parent drug from the enzymatic attack and so on, or sometimes to increase its activity, or sometimes to just I mean make the our parent drug more metabolically feasible and so on, to increase the activity. So this kind of a drug mixtures are very common in the medicinal chemistry. Many drugs you will see are given in combination, two drugs together and so like that. Now let us, so augmentin is some similar type of drug mixture. So now let us see what is the correct mixture of the augmentin. So it is the number B, amoxicillin and clavulanic acid. So this is the structure of the amoxicillin. Amoxicillin is a group of penicillin, okay, and this is the clavulanic acid, which is also, you can say, it has a structure similar to penicillin, but it has a oxygen group here rather than sulfur, and it does not have any side chain here in this position, but it has a beta lactam ring, and the particular stereochemistry is similar to the clavulanic acid. So clavulanic acid is a very important, I think, I mean, one of the very important components. Okay, so it was isolated from the clavuligerous by becomes in 1976. It has a weak and unimportant antibiotic activity, but it is a powerful and irreversible inhibitor of the most beta lactamases, which means that it is used as a sentry drug, is in combination with the traditional penicillin such as the amoxicillin in augmentin mixture. This allows the dose levels of amoxicillin to be decreased and also increases the spectrum of the activity. However, it should be noted that there are various types of beta lactamases. Although clavulanic acid is effective against most of these, it is not effective against all. Clavulanic acid is also administered intravenously with the ticarcillin as the timentin. So this is also important, that it is also administered intravenously with the ticarcillin as the timentin. Like augmentin, we also have this timentin. So question, the augmentin is very common one. So the question can be asked with the timentin also. So you better to remember it. Okay. So the structure of clavulanic acid was the first example of naturally occurring compound where the beta lactam ring was not fused with a sulfur containing ring. Instead, it is fused with the oxazolidine ring. So this is the oxazolidine ring, 5-member ring with the oxygen and nitrogen in 1,3 position. It is known as the oxazolidine ring. Okay, and the structure is also not is also unusual in that it does not have any acyl amino side chain. So as I have mentioned, it does not have a acyl amino side chain like this penicillin. Okay, so many analogs have been now made, and the essential requirement of the beta lactamase inhibitions are, so this is the SAR of clavulanic acid, which is also very important. Okay, so this is the oxazolidine ring, this 5-member ring with the nitrogen and oxygen. This is a beta lactam ring. There is no acyl amino chain here. The sulfur is replaced by oxygen here. Okay, and also this has a enol ether type of structure, where this has a Z stereochemistry. So a strain beta lactam ring is necessary. The enol ether is important. The Z configuration of the double bond of the enol ether, which is activity is reduced but not eliminated if the bond is E. No substitution as C6. So C6 we have no substitution. R stereochemistry at the position 2 and 5. The position 2 and 5 we have the R stereochemistry and the carboxylic acid group, which is also important. It is also that the 9-hydroxyl group is involved in the hydrogen bonding interaction with the active site. There is a 9-hydroxyl group, this one, which is involved with the hydrogen bonding interaction with the active site of beta lactamase enzyme. Clavulanic acid is a mechanism based irreversible inhibitor and can be classified as a suicide substrate. So these things are also important. Okay, that different type of enzyme inhibitor, the suicide inhibitor, I have already mentioned it for a couple of times, that enzyme inhibition is also a very important topic. Okay, the competitive inhibitors, non-competitive, uncompetitive inhibitors, reversible, irreversible inhibitors, suicide inhibitors, all the enzyme inhibitors are very important. And clavulanic acid is a very classic example of a suicide substrate, and the mechanism by which it inhibits the beta lactam, the beta lactamase is a mechanism based inhibition. Okay, the mechanism based irreversible inhibition, and the SAR of the clavulanic acid is also very important, which you need to remember. Okay, so now I will go to the next problem. Okay, so problem number 7, it says that the only DNA antisense molecule that has been approved as an antiviral drug is, options are nevirapine, number B is formivirsen, number C is darunavir, number D is zidovudine. The only DNA antisense molecule that has been approved as the antiviral drug is nevirapine, formivirsen, darunavir or zidovudine. The only DNA antisense molecule that has been approved as the antiviral drug will be nevirapine, formivirsen, darunavir or zidovudine. So already I have discussed earlier that the antiviral agents, there are different types of the antiviral agents which are important. Okay, different type of enzyme inhibitors. Vaccination is also one pathway which we have seen, but there is only one molecule in the market which is a DNA antisense molecule which is approved for as a antiviral drug, and the correct answer will be number B, formivirsen. Okay. So what is the antisense oligonucleotide therapy? So we have the DNA. DNA after transcription forms the mRNA, which after translation it forms the DNA as a disease associated protein. Okay. But in case of what happens in the oligonucleotide therapy, we have a antisense drug which is the oligonucleotide. Okay. So basically this is a oligonucleotide which acts as an antisense strand to the mRNA which is synthesized, and because of that RNase degrades the RNA and no translation happens, so no disease associated proteins are produced. So antisense therapy, formivirsen, is the first and so far the only DNA antisense molecule that has been approved as a antiviral agent. This is the structure of the formivirsen. You do not need to memorize it. I just have given you it for your understanding that how the antisense molecule looks like. It consists of the 21 nucleotide, as you can see, 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21. So as you can see, this G C G T T T G C T C T T C T T C T T G C G. So there are 21 total nucleosides are present. Okay. And these all with a phosphonothionate backbone rather than the phosphate backbone to increase the metabolic stability of the molecule. The drug blocks the translation of the viral RNA and is used against the retinal inflammation caused by the CMV in AIDS patients. Because of its high polarity, it is administered as an ocular injection, and which is known as the intravitreal injection. So mode of drug administration is also very important topic of our medicinal chemistry course. I have already discussed it for a couple of times, like what are the different modes of administration. As you I think you have remembered it, because we have learned different methods by which the drugs are administered. That there is oral way, intravenous way, sublingual way, sometimes the dermal injection, topical, then sometimes we can inject the drug to the spinal cord directly, and also ocular injection. So these are the different nasal, the through the inhaler and so on. So these are the different modes of the drug administrations, and we have already discussed it for a couple of time. So this particular formivirsen, it is so polar that it is administered as an ocular injection, which is known as the intravitreal injection. Okay. So now I will go to the next problem. So problem number 8, it says that statins works as what? Your options are number A, HMG-CoA synthase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthase inhibitor, number D, acetoacyl-CoA thiolase inhibitor. Statins works as which of the following? HMG-CoA synthase inhibitor, HMG-CoA reductase inhibitor, squalene synthase inhibitor, or acetoacetyl CoA thiolase inhibitor. Statins works as what? Of the which of the following? HMG-CoA synthase inhibitor, HMG-CoA reductase inhibitor, squalene synthase inhibitor, acetoacyl-CoA thiolase inhibitor. So the topic of statins, I have not discussed it earlier because we don't have it in our course in full, I mean, with much detail. But statins are a very important class of therapeutics, and in the very important topic of medicinal chemistry, we have introduced the statins for a few couple of times, but not in much detail. So now let me discuss it in with detail. So how the statin, what are statins, and how the statins work. So that we will discuss today. Okay. So basically, the correct answer will be HMG-CoA reductase inhibitor. So statins, they work as the HMG-CoA reductase inhibitor. So what is the HMG-CoA reductase inhibitor? So these are known as the HMG-CoA reductase inhibitors, also known as the statins, are a class of the lipid lowering drugs that reduce illness and mortality in those who are at a high risk of the cardiovascular disease. They are the most common cholesterol lowering drugs. Reduce the risk for the illness related to the atherosclerosis as with a varying degree of effect depending on the underlying risk factor and the history of the cardiovascular disease. Clinical practice guidelines generally recommend people start with the lifestyle modification through a cholesterol lowering diet and physical exercise. Mechanism: the HMG-CoA reductase pathway, which is blocked by the statins via inhibiting the rate-limiting enzyme HMG-CoA reductase. Statins act by competitively inhibiting HMG-CoA reductase. That rate-limiting enzyme is of the mevalonate pathway. Because statins have similar structure to the HMG-CoA on the molecular level, they will fit into the enzyme's active site and compete with the native structure, which is the HMG-CoA. This competition reduces rate by which the HMG-CoA reductase is able to produce the mevalonate. The next molecule in the cascade is eventually produces the mevalonate. The next molecule in the cascade that eventually produces the cholesterol. So this is also important that HMG, the statins, they are similar to the structure. Okay. So the statins are the they are competitive inhibitor of the HMG-CoA reductase, and because they have the similar structure with the native substrate, so then this they compete with it and fit on into the active site, and by which it inhibits the structure. So what are the examples of the statins? So these are the drugs which are which are marketed and FDA approved in various years. So as you can see, pravastatin is FDA approved in 1991. It has a molecular weight of 424.53, so 424.53. So this has this structure. This red group which is mentioned here is the pharmacophoric group of the statin. Then we have the simvastatin, which is approved in 1991, and it has a molecular weight of the 418.57. Then we have the fluvastatin. Okay, the fluvastatin also has this particular molecular weight of 411.47, and it was FDA approved in 1993. This is the structure of atorvastatin. Atorvastatin is a very widely used as a drug, which is also known as a Lipitor. Okay, it has a molecular weight greater than 500, as you can see, 558.64 other, even if this molecular weight is high, it is a very good drug, and this is the pharmacophoric group mentioned in the red. We have the rosuvastatin, which is FDA approved in 2003, and it has a molecular weight of the 481.54. We have the pitavastatin, which is approved in 2009, and it is molecular weight of 421.46. We have the mevastatin, which is a molecular weight of 390.51, and it is not FDA approved till now. It has in clinical phases. And lovastatin, it is FDA approved, okay, and it is in the 1987. It's at molecular weight of 404.54. So these lipid lowering drugs are very important, and the cholesterol lowering drugs are another very important clinical aspect, which was not discussed earlier in detail. Today also we will discuss a little bit about the statins in detail at the later of our today's class. Okay, so in detail we will discuss a little bit about the statins. Now let me go to the next question. So now we will go to our problem number 9. Okay. Problem number 9 says, which of the following is the correct order of the IC50 values for the following anti-agents against the human cancer cell lines? Okay, your options are number A, pyrimethamine, number B, trimethoprim, number C, methotrexate. Which of the following has the correct order of IC50 values for of the following anti-agents against the human cancer cell lines? So all these are the therapeutic agents, and you have to identify the correct order of the IC50 values of these agents against the human cancer cell line. Your options are number A greater B greater C, number B greater C greater A, number C is the B greater A greater C, number D is the C greater A greater B. Which of the following is the correct order of the IC50 values of the following anti-agents? So this is a kind of tricky question. You have to just read the question very carefully. You have to read the name of the therapeutic agents. All the therapeutic agents are for a particular, they have a particular clinical aspect. And if you can remember the clinical aspects, then you can easily answer this question. So many times the question can be asked to you, which are very direct in nature, and you can answer the direct question in very easily. But sometimes some questions can be asked to you which are not direct at all. Sometimes the question can be asked and which are indirect in nature. In this kind of tricky question, you have to read the question very carefully, and then you have to answer the questions, right? So this type of question, which is the problem number 9, which I have shown to you. So this is a this is a class a type of the tricky question. Okay, so now let us understand what is the question and then understand the answer. So the question was asked to identify the IC50 value, the correct order of IC50 value. So now the question can rise that if you do not know what is the correct what is the exact IC50 value, how to answer this kind of question. So the question, it asks that you have to identify it against the human cancer cell lines, right? So the pyrimethamine, it is a so first let us understand what is the correct answer. The correct answer is B greater N greater C. Okay. So one second. Yeah. So the correct order is then B greater C, B greater C, A greater than C. Okay. So why it is so? Basically, if you look at the options are given, so B is the trimethoprim. A is the pyrimethamine and C is the methotrexate. Among all these three, only methotrexate is an anti-cancer drug. It's a marketed anti-cancer drug, right? Anti-cancer agent. I have already mentioned it a couple of times. Okay, the trimethoprim is not anti-cancer. It is an antibacterial agent, and pyrimethamine it is an antimalarial agent. So since it is asked that, so if you know that is this is an anti-cancer agent, means it has the lowest IC50 value against the particular uh, our disease. I mean it has the lowest IC50 value particular against the human cancer cell. That is why it is a anti-cancer agent. Then the other also you have to just sometime you have to know the values a little bit, and you have to have some understanding. Okay, so first let us understand what is the IC50. So half maximal inhibitory concentration is a measure of the potency of a substance in inhibiting a specific biological or biochemical function. IC50 is a quantitative measure that indicates how much of a particular inhibitory substance, that is a drug, is needed to inhibit in vitro a given biological process or biological component by 50%. The biological component could be an enzyme, cell receptor, microorganism. IC50 values are typically expressed as the molar concentration. All these molecules are acting as drug. For example, pyrimethamine acts on the plasmodium, and that is reflected in the IC50 values. The IC50 is the inhibitory concentration required to inhibit 50% of the activity. So IC50 value for the plasmodium is 0.05 microgram per milliliter. For the bacteria it is 250, for mammal is 70. So you see that it is extremely specific for the plasmodium. So pyrimethamine is used as an anti-malarial agent because plasmodium is the causative organism for malaria. So this is the antimalarial agent. Trimethoprim is a bacterial dihydrofolate reductase inhibitor. The IC50 value for bacteria is 0.5, for plasmodium is 7. So it is 10. It is a 10 about 14 times higher. Remember, lower the IC50 value, stronger is the potency. For mammal, it's totally ineffective because IC50 value is 26,000. So one can use trimethoprim as antibacterial agent. For methotrexate, IC50 value is 0.01 for bacteria, for plasmodium, and for mammal is 0.02. So it can act as an antibacterial agent, antiplasmodium agent. Also it can used as in the dihydrofolate reductase in mammals. It kills the cells of the human body, specifically the cells which grows fast, that is the cancer cells. So methotrexate is used as an anti-cancer agent. This is your antibacterial. So this is your antibacterial agents. Okay, that is antimalarial agent. So you can now target DHFR specifically. The three phases of drugs have been developed, and these are applied on bacteria, and one to parasitic, and one to the human. So these three are available in the market. Okay. So that is how this type of questions are answered. So sometimes the questions are tricky, as I have already mentioned. It's not like always the question will be given very straightforward manner, but sometimes the question can be given which are tricky in nature. So you have to just read the question, and you have to use your understanding, or you have to use your own judgment to answer this kind of question. So now I will go to the next problem. Okay. Now let me go to the problem number 10. So problem number 10 says that oseltamivir is an dash agent. Your options are number A anti-influenza, number B anti-herpes virus, number C antiretroviral, and number D antifungal. Oseltamivir is an which kind of agent? Anti-influenza, anti-herpes virus, antiretroviral, or antifungal? Oseltamivir is an dash agent. You have to identify the correct option. Number A anti-influenza, then anti-herpes virus, antiretroviral, or antifungal. Which of the following will be the correct answer regarding oseltamivir? So sometimes the students ask that there are so many drugs and so many therapeutic agents that do they need to remember all those things, or do they need to memorize all those things? So there are some tricks by which you can remember which kind of drug. If the name is given which kind of drug it is, you can make a rough guess. Okay, but these things you need to remember. So there are few strategies. I will discuss this in the later of our course, when I will discuss various therapeutic agents like antibacterial, antiviral, and so on. So then I will discuss this particular thing. So this particular question, which is asked about the oseltamivir, so you have to now identify which is the what is the clinical aspect and what type of drug this oseltamivir is, right? So now let us see what kind of agent this oseltamivir is. So okay, so the oseltamivir is a it is an anti-influenza agent. Okay, so oseltamivir is an anti-influenza agent. So now let us see the clinical aspect regarding it. So this is the structure of the oseltamivir, which is also known as the Tamiflu. So oseltamivir is used for the prevention and the treatment of the influenza caused by the influenza A and B viruses. It is on the complementary list of the World Health Organization's list of essential medications, the most important medication used in a basic health system. As that of December 15, 2010, the WHO, or the World Health Organization, has reported 314 samples of the prevalent 2019 H1 pandemic H1N1 flu tested worldwide have shown resistance to the oseltamivir. The vast majority of mutation conferring resistance are single amino acid residue substituent, the histidine 274 tyrosine in N1 in the neuraminidase enzyme. In 2011 meta-analysis of 50, that is, have found a pooled incidence rate of the oseltamivir resistance of the 2.6%. Subgroup analysis detected higher rates among the influenza A patients, especially H1N1 subtypes. Nevertheless, oseltamivir is a very important drug which is used against the influenza A and B viruses, and it is a very important antiviral drug in that case. Okay, now I will go to the next problem. Problem number 11. It says which of the following statements is false regarding the interferons. Your options are: number A, they are classified as a broad spectrum antiviral agents. Number B, they are small natural proteins. Number C, beta interferons are generated from lymphocytes. Number D, immunomodulators are used to induce the interferon production. Which of the following statements is false regarding interferons? Number A, they are classified as a broad spectrum antiviral agent. Number B, they are small natural proteins. Number C, beta interferons are generated from lymphocytes. Number D, immunomodulators are used to induce the interferon production. Which of the following statements is false regarding the interferons? So interferons are very important and comparatively some offbeat topic, but it is a very important antiviral, antiviral agents. In case of the antiviral drug discovery, interferons, they take actually a very large space. Okay, so earlier I didn't get the chance to discuss about interferons, but today I thought it is better to us to learn something about the interferons also. I will wait for a minute and then I will show you the correct answer, and then I will discuss about the interferons in detail. So whatever the topics which I am discussing today are mainly from your course content itself. Some topics I have not get the chance to discuss in properly in earlier, and some topics which are some a little different, but these are very important also. So this I thought to just discuss it. So now let us understand what are the interferon and what are the clinical aspects of the interferon. So basically, interferons, so the statement which is false is number C, which says that beta interferons are generated from the lymphocytes. Okay, So now let us understand. So interferons are small natural proteins that were discovered in 1957 and which are produced by the host cells as a response to the foreign invaders. Once produced, the interferons inhibit the protein synthesis and the other aspects of the viral replication in the infected cells. In other words, they shut the cell down. This can be described as the intracellular immune response. Administering interferons to patients has been seen as a possible approach to treating the flu, hepatitis, herpes, and colds. There are several interferons which are named according to their source. Alpha interferons from the lymphocytes, beta interferons from the fibroblasts, and gamma interferons from the T cell. Alpha interferon, also known as the alpharon or IFN alpha, is the most widely used of the three types, and in the past it was difficult and expensive to isolate the interferons from their natural cells. But the recombinant DNA techniques allow the production of the genetically engineered interferon in larger quantities. Recombinant alpha interferon is produced in three main forms. The alpha 2A and alpha 2B are the natural forms, and the alpha con 1 is the unnatural form. They have proved successful therapeutically but can have serious toxic side effects. At present, alpha interferon is used clinically against the hepatitis B infections. It is also used with the ribavirin against the hepatitis C infections. Interferon production in the body can also be induced by agents known as the immunomodulators. One such example is the avridine, which is used as the vaccine in the vaccine adjuvant in the treatment of animal diseases such as foot and mouth. Imiquimod also induces the production of alpha interferon as well as other cytokines that stimulate the immune system. It is effective against the genital warts. So let us now understand how the this antiviral action of interferon, how it works actually. So we have a viral RNA in a virus infected cell. So how the viral infection happens? So this is also very important, how the viral infections happen. Okay. So in our earlier lectures, when I was discussing about the this antiviral agents, I have discussed that how the viral infection happens, what are the steps of the viral infection, like the the attachment, then the penetration, followed by the replication, and then the synthesis of the genetic material, and after that the release. And these things are very important, along with the structure of the virus, especially the HIV virus, how the structure, how it looks like, what are the various components, what are the various roles of the various components. So these things which are important, which I am not discussing today, but I have discussed it earlier. There is a presentation, there is a recording which you can refer to while reading about these things. Okay. So now let us go and discuss and see what is the how the antiviral effect of the interferon it works actually. So basically we have a virus infected cell, right? So this is the antiviral, so virus infected cell. So the first one is the viral RNA from an infecting virus enters the cell. Okay, so first the viral RNA goes to the cell, and then the step 2 is the infecting virus replicates into new viruses. So the infected virus, now it replicates to the new viruses, as you can see here. And step number 3 is very important. Infecting virus also induces the host cell to produce interferon mRNA, which is the IFN mRNA. Okay, and which is translated into the alpha and beta interferon. So there is the IFN mRNA, as you can see, and which induces the viral infected cell to produce the alpha beta interferon, which is the step 3. Now the fourth step is another very important step. Interferon released by the virus infected host cell bind to the plasma membrane or the nuclear membrane receptors on the uninfected neighboring host cell. So as you can see here, this interferon released by this virus infected cell is now binding to the plasma membrane of the nuclear membrane of the receptors on the uninfected neighboring cells. This is an uninfected neighboring cell. Okay, and inducing them to synthesize the antiviral protein or AVP. So this, after introduced to cell, it activates the cell for synthesizing the antiviral proteins. Okay, this include the oligoadenylate synthesis and protein kinase. So new viruses released by the virus. So not only interferons will be released by the virus infected cell, also new viruses will also be released by the virus infected cell. So now new viruses, when they try to when the new virus and its RNA I mean it is inserted into the non-infected cell, where the antiviral proteins are already present due to the interferon, what will happen? This new viruses, for this ABPs or this antiviral proteins will degrade the viral RNA, mRNA, and inhibit the protein synthesis and thus interfere with the viral replication. Okay, so these are the 6 steps with the figure. I think you have understood this. First step is the viral RNA introduction to the virus infected cell, then the replication of the virus, and this series will trigger the host cell to synthesize or the produce the interferon. The interferon will be released so as the new virus particles. Interferon will induce the nearby host cell to for synthesizing antiviral proteins, which once the viral RNA will go inside the host cell of the healthy cell, then this antiviral proteins will degrade the mRNA and it will interfere with the viral replication. So that is how the antiviral effect of the interferons happen. Okay. So the interferons, that is how the things happens with the interferons. So the statements, as you can see, the question. So what were the statements? So in the statement number C, it said which said that the beta interferon is produced from the lymphocyte. It is false, because clearly I have mentioned that alpha interferons are produced from the lymphocyte. So interferons are indeed classified as a broad spectrum antiviral agents. They are small natural proteins, and they are not so beta interferon are not generated from lymphocyte. Alpha interferons are generated from the lymphocyte. Beta interferons are generated from the fibroblast, and immunomodulators are used to induce the interferon production. It is also correct. Okay. Now I will go to the next problem, and this is the last problem which we will discuss today. And so problem number 12, it says which one of the following activities occurs following the stimulation of the H2 receptors. Your options are number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, and number D, enhanced secretion of the hydrochloric acid in the stomach. Which of the following activities occurs following the stimulation of the H2 receptors? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of the hydrochloric acid in the stomach. So the problem number 12 now we will discuss. Okay. So which of the following activities occur following the stimulation of the H2 receptors? So the following the activities that occurs following the stimulation of H2 receptor will be number D, enhanced secretion of the hydrochloric acid in stomach. So H2 receptor is basically the histamine receptor. So there are 4 types of histamine receptors: H1, H2, H3, and more recently H4. So G protein coupled receptor activation of H1 cause the contraction of smooth muscles in the gut, the uterus, and the bronchi. Contraction of the bronchi leads to the asthma. Stimulation of H1 receptors on the smooth muscles in fine blood vessels cause the muscle relaxation, and the resulting vasodilation may result in severe fall of the in the blood pressure. In CNS, H1, H2 receptor predominantly localized on the postsynaptic membrane. H3 receptors appear on the function predominantly as the presynaptic receptors, possibly as the histamine autoreceptors, and has been detected in some peripheral organs. H4 receptor exhibit very restricted location in the intestinal tissue, spleen, and the immune active cells, and for example T cells, suggesting the therapeutic potential of the H4 blocker in allergic and inflammatory diseases. So H1 versus H2 receptors. So H1 receptors, what is the definition? Histamine receptors that couple with the GQ11 stimulation, stimulating the phospholipase lipase C. The location is the periphery of the body, adrenal medulla, vascular endothelium, heart and central nervous system. The antagonists are the diphenylhydramine, the loratadine, cetirizine, cetirizine, fexofenadine, and clemastine. Ileum contraction, modulates circadian cycle, itching, systematic vasodilation, bronchoconstriction, allergy-induced asthma. These are the functions for the H1 receptor. For the H2 receptor, the definition: histamine receptor that interact with the GS to activate the adenyl cyclase. So, the location is the CNS or the central nervous system, and the periental cells located in the stomach. Antagonists are the ranitidine, cimetidine, famotidine, and nizatidine. And the function: speeds up the sinus rhythm, stimulation of the gastric acid secretion, smooth muscle relaxation, inhibit the antibody synthesis, T cell proliferation, and cytokine productions. Okay. So these were the questions which I have to discuss today in the summary session. Okay. So now what I will do is, after discussing the question, I have taken a few few topics which I am going to discuss today. Okay, so these topics are basically very important, and these topics are related to the various therapeutic effects. So I will start with the antibacterial agent, then I will discuss about the antiviral agent, cholesterol lowering agents, also with the antidepressant agents and antifungal agents a little bit. I will discuss about anti-cancer agents, and also I will discuss about the cholinergic systems. Okay, so why I have chosen these topics for discussing in today's summary class is, the reason is very obvious, because because most of the students are asking me one thing. They have mailed me for a couple of time, and also they have asked during the classes also, that they do not understand which topics to read, and which topics to do not read, which are the important topics. And they are mostly confused that there are so many therapeutic agents, there are so many drugs, and there are so many things to how to remember all these things. Okay, so I will just summarize the things so for your understanding, and also I will just show you some things. I will show you how to remember some therapeutics based on their names and on so on. So these are little tricks which I will just share with you. So first topic which I am going to discuss is the antibiotics. So antibiotics and all the antibacterial agents is a first class of therapeutic agents which we started discussing, right? So the antibiotic classes which are very important are the penicillins, cephalosporins, quinolones, macrolides, and tetracyclines. Okay, so penicillins. So penicillins has the penicillin. So there are several generation of the antibacterial agents. Any class will have several generations. Okay, because in previous, so when first generation is synthesized, there are several different problems in the first generation. Based on the other generations are synthesized. So for penicillin, the first generation is the penicillin G, so generation 2 is the amoxicillin, then generation 3 is the ticarcillin, generation 4 is the piperacillin. Okay, so these are the, so what you have to remember is what is the particular the common structure of a particular antibiotic class, and also what is the mechanism of action of that particular class. So this is the penicillin. What is the role of penicillin? How it inhibit the bacterial growth? You need to remember it. Okay, and this is the basic structure which you need to remember. There is a beta lactam ring fused with a thiazolidine ring, and these are the SAR which I have already mentioned to you, right? So then we have, so the name of penicillin, so as you can see, amoxicillin, ticarcillin, piperacillin, so it has the cillin suffix in all the components. So whenever there is this suffix, you have to identify this is the penicillin class of compound. For the cephalosporin, we have this prefix of the ceph, as you can see, cephalotine, which is the first generation. Then second generation is the cephioximine, ceftazidimine, which is third generation, and cefepime is the fourth generation. We have the quinolones. Quinolone is also very crucial class of antibacterial agent. As you can see, this has a quinolone ring, which is the pharmacophore, and with the other rings, and there there is this carboxylic acid group in the one. This 3 position is also very crucial for the activity of the quinolones. Okay, now, so all the classes along with their names, their therapeutic, I mean the therapeutic areas, and also the SAR is very important, which you need to remember along with the examples. Okay, so nalidixic acid, ciprofloxacin, levofloxacin, and moxifloxacin. These are the quinolone class of antibiotics. So oxazine is the suffix which you need to remember. We have macrolide. Like macrolide, the structure is little bit I mean confusing. I mean little bit tough, I know. You won't be asked to write the structure, okay, but if you the question is given with the structure, you have to you I know I want you to identify that it is, yes, it is a macrolide. That much you need to do, okay. And so this macrolide, we have the erythromycin, calithromycin, telithromycin, and mycin is the particular suffix which is you have to remember. The thromycin, as you can see, all have this mycin. And we have the tetracycline, which has the cycline suffix also. Tetracycline, four cycline rings are fused with each other in the case in the tetracycline, okay. So now what are the first generation is the oxytetracycline, then doxycycline, tricycline. So these are the tetracycline class of the antibiotics. So these are the most crucial class of the antibacterial agents, along with the different generation of the drugs, and also with their common structure. So the things you need to remember is the mechanism of action, the common structure, especially of these 5, okay, and also their examples. So not only this, which you have to remember, the mechanism of action. Now, how to remember the mechanism of action? The easiest way to remember the mechanism of action is with this figure. Okay, this is the complete figure where the mechanism of actions are mentioned. So first, bacterial cell, it has a cell wall. So cell wall synthesis can be inhibited by couple of antibacterial agents. For example, cycloserine, vancomycin, bacitracin, penicillin, cephalosporins, monobactams, carbapenems. So these are the components which inhibit the cell wall synthesis. For folic acid metabolism, trimethoprim, sulfonamides are there. Okay, so they inhibit the folic acid metabolism. So folic acid metabolism is a very interesting pathway where para-aminobenzoic acid is converted to DHA, which is the dihydrofolic acid, to the THF, which is tetrahydrofolic acid. Okay, so sulfonamide, they inhibit this first step, which is the conversion of PABA to DHF, because sulfonamides, they act as the competitive inhibitors, which the structure is similar to the substrate para-aminobenzoic acid. That's how it inhibits this enzyme which is catalyzing this pathway, okay, of the PABA to DHF. I have already discussed it. And trimethoprim is inhibit the conversion of DHF to THF. Then we have the cytoplasmic membrane structure and function, polymyxin and actinomycin. They inhibit this work. All these things I have discussed in our courses earlier also. And lipid biosynthesis is inhibited by the plantancimycin. Then protein synthesis, the tRNA is inhibited by the mupirocin and puromycin. Protein synthesis can be inhibited by inhibiting the ribosomes also. So in the bacterial cell, the prokaryotic ribosome has 2 subunit. One is the 50S and one is the 30S subunit. Okay, so the protein synthesis can be inhibited by the 30S subunit inhibitors, which are the tetracycline, streptomycin, spectinomycin, gentamicin, kanamycin, amikacin, and nitrofurans. Okay, and protein synthesis 50S inhibitors can be also they are like erythromycin, the macrolides, the chloramphenicol, clindamycin, and lindamycin. These can also inhibit the protein synthesis by inhibiting the 50S of the ribosome. Then we have the mRNA, the genetic materials, which are also can be inhibited as the antibacterial agent, can also inhibit the genetic material. Like we have this DNA directed RNA polymerase inhibitor like rifampicin and streptoviracins, and we have the RNA elongation inhibitor like actinomycin. And DNA guidase is a very crucial enzyme for the bacterial replication. In bacterial replication, these are the class of the topoisomerase enzymes. Okay, quinolones is the class of the drug which basically like nalidixic and ciprofloxacin. Also novoviacin, they act on the DNA guidase. So these aspects are these are the things which are related to your antibacterial agents. Earlier also I have discussed about the antibacterial agents. So whenever so not only the summary class, so earlier classes when the antibacterial agents are in the week 10, if I am not wrong, it is in the week 10. Okay, now in the particular week 10 I have already discussed a lot about this antibacterial agents, how they work, and what are the clinical aspects of different antibacterial agents. In detail I have have discussed, you can go through that particular slide also and along with the particular video. So that is the summarization of the particular your antibacterial agents. Okay, that is how I just if you remember this particular figure, I think it will be easier for you. Now let us understand about the antihypertensive agents or antihypertensive drugs. Okay, so the antihypertensive drugs, so what the mechanism by which the blood pressure is increased in our body, that mechanism is also very important. Today I am not going to give the detail about how our blood pressure is how the renin angiotensin system works, because I have already mentioned it for a couple of times, how the renin angiotensin system works, what are the various aspects of the renin angiotensin system, what are the enzymes that are involved. Okay, that all those things I have already discussed for a couple of time earlier in our course, so you can go through the slides to understand these things. So okay, so now let us understand different classes of antihypertensive drugs. Here again, the mechanism of action, remembering the mechanism of action and the example is very important. So first is the diuretics. So diuretics, also called the water pills, are the medications designed to increase the amount of water and salt expelled from the body as the urine. They are often prescribed to help to treat the blood pressure. Furosemide, sold under the brand name of Lasix, among the others, is a loop diuretic. This is the structure of furosemide. Medication used to treat the fluid buildup due to the heart failure, liver scarring, or kidney disease. It may be also used for the treatment of the high blood pressure. Furosemide was patented in 1959, and it is a World Health Organization's list of the essential medication. Amiloride is a very commonly used diuretic. Okay, so it is sold under the trade name of the Midamor, among the others. It is a medication typically used with other medications to treat the blood pressure or swelling due to the heart failure or the liver cirrhosis. SE inhibitors is a very important class of the antihypertensive agents. The angiotensin converting enzyme inhibitor or SE inhibitor is a class of medication used primarily for the treatment of the high blood pressure and the heart failure. They work by causing the relaxation of the blood vessels as well as decrease in the blood volume, which leads to lower the blood pressure and decreased oxygen demand in the heart. SE inhibitors inhibit the activity of the angiotensin converting enzyme, an important component of the renin-angiotensin system, liable to convert the angiotensin 1 to angiotensin 2 and hydrolyze the bradykinin. Therefore, ACE inhibitors decrease the formation of angiotensin 2, a vasoconstrictor, and increase the level of the bradykinin, a peptide vasodilator. This combination is synergistic in lowering the blood pressure. Captopril, sold under the trade name of Capoten, is an angiotensin converting enzyme or ACE inhibitor used for the treatment of hypertension in some types of congestive heart disease. Okay, so this is the structure of the captopril. So again here you can see the pril. This pril, whenever there is this suffix, you will understand it is a, you have to figure out that it is a angiotensin converting enzyme inhibitor or ACE inhibitor. Then we have the 81 blockers. So bradykinin angiotensin 2 receptor blockers, formerly angiotensin 2 receptor 1 type 1 antagonist, also known as angiotensin receptor blocker, angiotensin 2 receptor antagonist, or 81 receptor antagonist, are a group of pharmaceuticals that bind to and inhibit the angiotensin 2 receptor type 1 or 81, thereby block the arterial contraction and the sodium retention effect in the renin angiotensin system. Their main uses are the treatment of the hypertension, high blood pressure, diabetic nephropathy, the kidney damage due to diabetes, and the congestive heart failure. They selectively block the activation of the 81 receptor, preventing the binding of angiotensin 2 compared to the ACE inhibitors. These substances are 81 receptor antagonists, that is, they block the activation of the angiotensin 2 81 receptors. 81 receptors are found in the smooth muscles of the vessels of the cortical cells of the adrenal gland and adrenergic nerve synapses. Blockage of the 81 receptors directly causes the vasodilation, reduces the secretion of the vasopressin, and reduces the production and the secretion of the aldosterone, among the others. The combined effect reduces the blood pressure. Losartan, irbesartan, omnesartan, candesartan, valsartan, fimesartan, azilosartan, they also include the tetrazole group. So these are the examples of the 81 blockers. Then we have the beta blockers. So beta blockers are comparatively newer classes of antihypertensive agents. So these are the examples like atenolol or tenormin is a beta blocker that affects the heart and circulation, the blood flow through the arteries and veins. Atenolol is used to treat the angina, chest pain, and hypertension, the high blood pressure. Atenolol is also used to lower the risk of the death after the heart attack. Nebivolol is a novel, highly cardioselective beta blocker with the antihypertensive efficacy similar to that of the other beta blockers, but with tolerability better than other agents in its class, which may be permit nebivolol to be the most used widely and effectively than other beta blockers. Yes, nebivolol is a very important and the most crucial beta blocker, actually the most used one. And metoprolol is a beta blocker that affects the heart and circulation, blood flow through the arteries and veins. And metoprolol is used to treat the angina, the chest pain, and the hypertension. It is also used to lower your risk of the death needing the hospitalized after the heart failure. So these are the different your antihypertensive agents which are used. You have to understand that one thing is that not all the antihypertensive use agents will be used with the particular patient. So why, now how the doctors they choose that which type of drug is given to a particular patient? So it depends on the various things like the age of the patient, the gender, age, the clinical history of the particular patient, and the symptoms, like whenever there is a irregular heartbeat, or there is a irregular heartbeat, or in case of the anxiety and those things, beta blockers are preferred because they actually help to regularize the chest pain, and also the with the irregular heart system. So that's time beta blockers are used. Some patients with the either age and all those things, this particular antihypertensive agents are used. Okay, so this depends on the many things, and one of the which I have mentioned. So these are the antihypertensive agents which are very important, and you need to remember this antihypertensive agents. Okay, now I will go to the next topic. Now let us discuss the next therapeutic topic. Okay, so again here also mechanism of actions is very important. You need to remember the mechanism of actions for each and every class along with the examples. Okay. Now antifungal agents, let us discuss about little bit about antifungal agents, because we didn't get a chance to discuss about the antifungal agents with a very detail. Okay, so antifungal drugs are the medication deployed in the treatment of the mycosis, the fungal infections of the skin. Now as of 2010, the fourth most common disease in the world, affecting around 1 billion of people. So mycosis is a fungal infection of animals including the human. Mycosis are the common, and a variety of environmental and physiological conditions can contribute to the development on the fungal diseases. Okay, so what are the different antifungal drugs that are used? So imidazoles. So basically azoles are azoles, the compound which are the azoles, they actually have a very good prevalence in the antifungal drug discovery. Azoles mean it will have the 5-member ring which has the 2 or 3 nitrogen. So these are known as the azoles. Okay, one or more. So basically imidazole means it has a 5-member ring which has a nitrogen in 1 and 3 position, and triazole at the nitrogen contained in the 5-member ring which has 3 nitrogen, it can be either 1 to 3 position or 1 to 4 position. Imidazole, what are the imidazole containing antifungal drugs? These are the potrimazole, oxyconazole, miconazole, iconazole, ticonazole, tioconazole, and ketoconazole. The triazole containing, we have the fluconazole, itraconazole, tarconazole, voriconazole, isavuconazole, and posaconazole. Okay, and these are the azole antifungal. What is the mechanism of action? They inhibit the lanosterol 14 alpha demethylase, the enzyme required to convert the lanosterol to ergosterol. Then we have the griseofulvin. This inhibits the mitosis in the dermatophytes. It is ineffective when applied topically. We have the echinocandis, which is this caspofungin, anidulafungin, and micafungin. This inhibit the cell wall synthesis by targeting the glucans 1,3 beta glucan synthase. And we have the flucytosine pyrimidine analogs converted into the 5-fluorouracil by the fungal enzyme cytosine deaminase, active against the yeast infections. And then polyenes, the natamycin, amphotericin B, nystatin, We have the allyl amines, that amyrolophen, natifene, and terbinafine, which inhibit the squalene epoxide. So these are the various groups of the antifungal agents along with the examples and their mechanism of action. So one class which is very important, which is the triazole containing antifungal agent. For example, fluconazole. This is the structure of fluconazole, which has 1 to 4 triazole ring. Voriconazole also have 1 to 4 triazole, as you can see, 1, 2, and this 3 position is carbon and the 4 position is also nitrogen. And albaconazole is also 1 to 4 triazole containing antifungal drugs. So triazoles are the newer, less toxic and more effective antifungal agent. We have fluconazole, itraconazole, albaconazole, posaconazole, voriconazole. Like other imidazole and triazole class of antifungals, fluconazole, let us discuss about the fluconazole a little bit, because it is a very important antifungal agent. Fluconazole inhibits the fungal cytochrome P450 enzyme 14 alpha demethylase, mammalian demethylase activity is much less sensitive to fluconazole than the fungal demethylase. This inhibition prevents the conversion of the lanosterol to ergosterol, an essential component of the fungal cytoplasmic membrane, and subsequent accumulation of the 14 alpha methyl sterols. Fluconazole is an inhibitor of the human cytochrome P450 system, particularly the isozyme CYP2C19, CYP3A4, and CYP2C9 a lesser extent. Okay. And in theory, therefore, fluconazole decreases the metabolism, increases the concentration of any drug metabolized by the enzymes. In addition, the potential effect on the QT interval increases the risk of the cardiac arrhythmia if used concurrently with other drugs to prolong QT interval. So this was about the antifungal drugs. Antifungal drugs, we didn't discuss it earlier in detail, but I thought it is a very good option for us to discuss it in a summary class, because antifungal drugs are also very important class of therapeutics, which we didn't get the chance, but since we have the summary class, I thought it will be better if we discuss it a little bit. Okay. So so here also you need to remember the names of the drugs along with their application, the clinical aspect, the mechanism of action, and also their examples and the mechanism of action. Structure, if you can remember the structure, it is fine. If you cannot remember the structure, it is also fine, because yes, the structures are sometimes complicated and you may not remember all the structures. So now let us discuss about the antiviral drugs. Already I have discussed a lot about the antiviral drugs, but basically now I will discuss about the HIV a little bit. So HIV is the most common type of human immunodeficiency virus. It attacks your body's immune system. The virus destroys the CD4 cells. The cells help your body to fight infections. HIV-1 can severely damage your immune system and lead to the acute immune deficiency syndrome, or AIDS. CD4 cells are white blood cells and that play important role in the immune system. Your CD4 cell count gives you an indication of the health of your immune system, your body's natural defense system against the pathogen, infections, and illness. Currently, there is no cure of HIV or AIDS. Once you have the infection, your body can get rid of it. However, there are many medications that can control the HIV and prevent the complication. These medications are called the antiretroviral therapy, or ART. So what are the various therapeutics which are used as the antiviral agents? Let us discuss it a little bit. Already I have discussed about the antiviral agents in detail a lot of the time, but let us discuss it a little bit also again. So reverse transcriptase inhibitors. So reverse transcriptase, or RT, is an RNA dependent DNA polymerase that utilizes a strand of RNA to synthesize double-stranded viral DNA. This mechanism helps the retroviral gene to get incorporated into the host cell's gene. Various RT inhibitors are developed which are on the basis of the binding site and clinical chemical class of the compounds are classified into the NRTIs and nucleoside reverse transcript inhibitors, and then NNRTIs, the non-nucleoside reverse transcriptase inhibitors. So what are the NRTIs and NNRTIs? So non-nucleoside reverse transcriptase inhibitors. Among the HIV RT inhibitors, non-nucleoside reverse transcriptase inhibitors, or NNRTIs, have gained a definite place due to their unique antiviral potency, high specificity, low toxicity in antiretroviral combination therapies used to treat HIV. NRTIs, the nucleoside reverse transcript inhibitors, while NRTIs and NNRTIs are effective at the terminating the DNA synthesis and HIV replication. Nucleoside reverse transcriptase inhibitor, or NRTIs, inhibit the reverse transcription by causing the chain termination after they have been incorporated into the viral DNA. This was the first group of antiretroviral agent that to be used against the HIV. NRTIs lack the 3' hydroxyl group on the deoxyribose moiety. As a result, following the incorporation of the NRTIs, the next incoming deoxynucleotide cannot form the next 5' 3' phosphodiester bond needed to extend the DNA chain. Thus, when the NRTIs is incorporated, viral DNA synthesis is halted, a process known as the chain termination. All NRTIs are classified as the competitive substrate inhibitors. Okay. There are protease inhibitors also. Proteases are the enzyme that cleave the protein molecule into the smaller fragments. HIV protease is vital for both the viral replication within the cell and to release the mature viral particles from the infected cell. Various protease inhibitors are developed for the HIV treatment. Some of the FDA approved drugs are the class of the saquinavir, indinavir, as atazanavir, ritonavir, nelfinavir, amitriptyline, and lopinavir, and tenofovir. So there is a vir if the suffix, so you have to understand that this is the antiviral agent. So not only these are the important antiviral agent, there are several other antiviral agents which I have discussed earlier. Okay, we have discussed earlier about the interferons in today's class itself. Earlier we have discussed about the integrase inhibitors, how they act, okay, how they behave. Protease inhibitor, why they are important, what is the relevance of the protease in the antiviral drug discovery. All these things I have discussed in detail. You will find these things in the week 11 course content, where I have discussed about the antiviral agents in detail. So you also, along with the summary session PPT, you have to, and the video, you have to go through the week 11 one also. There you will find all the details about this antiviral agents and antiviral drugs. Okay. Now let me go to the next slide. Okay, so now I will go discuss about the next topic. So the next topic which we are going to look at is the cholesterol lowering drugs. Already we have discussed it a little bit, that the statins or the HMG CoA. So there are different classes of cholesterol lowering drugs, but today we will discuss about the statins. Okay, so statins are the class of the drugs often prescribed by the doctors to help to lower the cholesterol levels in the blood. By lowering the levels, they help to prevent the heart attacks and strokes. Studies show that in certain people, statins reduce the risk of heart attack, stroke, and even death after from the heart diseases by about 25 to 35%. Statins are also known as the HMG CoA reductase inhibitor. Earlier, I have discussed it about it, and today next I will also discuss about it. So statins are the class of drug often prescribed by the doctors. So this is the lovastatin sold under the brand name of the Mevacor. Okay, among the others, it is a statin medication to treat the high blood cholesterol and reduce the risk of the cardiovascular disease. Lovastatin, a compound isolated from the Aspergillus staius, was the first statin to be marketed. Simvastatin, sold under the brand name of Zocor, among the others, is a lipid-lowering medication. It is used along with the exercise, diet, and weight loss to decrease the elevated lipid level, also used to decrease the risk of the heart problems in those at a high risk. Simvastatin was patented by Merck in 1980 and came into the medical use in 1992. It was on the World Health Organization list of the essential medications. So there are other cholesterol lowering drugs also are present. Atorvastatin or Lipitor is a lipid lowering drug included in the statin class of medications. By inhibiting the endogenous production of cholesterol in the liver, statins lower the abnormal cholesterol and the lipid levels, ultimately reduce the risks of the cardiovascular disease. More specifically, statin medications competitively inhibit the enzyme hydroxymethylglutarate coenzyme A or HMGA reductase, which catalyzes the conversion of HMGA to mevalonic acid. This conversion is critical in metabolic reaction involved in production of several compounds involved in the lipid metabolism and transport, including the cholesterol, low density lipoprotein, sometimes referred to the bad cholesterol, and a very low density lipoprotein or VLDL. Rosuvastatin or Crestor is used along with the proper diet to help to lower the bad cholesterol and fats, such and raise the good cholesterol in the blood. This is the structure of atorvastatin. This is the structure of rosuvastatin. Then pitavastatin is a member of the blood cholesterol lowering medication class of statins. Like the statins, it is an inhibitor of the HMG CoA reductase, and the enzyme catalyzes the first step of cholesterol synthesis. But it was patented in 1987 and approved for the medical use in 2003. It is available in Japan, South Korea, and India. In US, it has received the FDA approval in 2000. 9. Okay, so this thing is also very important. One more point which I remember, that this drug, not only the drugs are important, the regulatory bodies are also important, like FDA approval is necessary for a drug to enter the US market. In India we have the FSSI, and also I have discussed it in, I think in our first class itself I have discussed about the various governing body. Europe has its own, Japan has its own, but FDA is the highest form, because FDA is the worldwide, the Food and Drug Administration of administration. So this is the most crucial regulatory body which checks if the drug is or which takes care of all the approval of the drugs and so on. So this is very important. So not only the cholesterol lowering drug, how the mechanism of cholesterol lowering is important, not only that, it is also important that this is also related with your enzyme inhibition. Now let us go to the next topic, antidepressants. So antidepressants also I have discussed it a little bit, because I have discussed about the myo inhibitors but not in detail. So let us discuss about the antidepressants also. So what are the antidepressants and how they work? So antidepressants are the class of drug that reduce the symptoms of the depressive disorders by correcting the chemical imbalances of the neurotransmitters in the brain. Chemical imbalances may be responsible for the changes in mood and behavior. Neurotransmitters are vital as they are communication link between the nerve cells of the brain. Neurotransmitters reside within the vesicles found in the nerve cell, which are released by one nerve and taken by other nerve. Neurotransmitters not taken up by other nerve is taken up by the same nerve that released them. This is process known as the reuptake. The prevalent neurotransmitter in the brain specific to the depressions are the serotonin, dopamine, norepinephrine, or also known as the noradrenaline. In general, antidepressants work by inhibiting the reuptake of the specific neurotransmitters, hence increasing their levels around the nerves within the brain, such as the selective serotonin reuptake inhibitors. Okay, antidepressant that will affect the serotonin levels in the brain. Monoamine neurotransmitters are neurotransmitters that, and a neuromodulator that certain amino acid is connected with the aromatic ring by two carbon long chain, like serotonin, dopamine, epinephrine, norepinephrine. So these are the examples. About the neurotransmitter, I have discussed about the neurotransmitters earlier. Okay, and also I have discussed about the we have discussed about the antidepressant. Only one class of antidepressant which I have discussed about is the myo inhibitors, and but the most common antidepressant drug is the serotonin, selective serotonin reuptake inhibitors, or SSRIs. Okay, for example are the fluoxetine, fluvoxamine, citalopram, sertraline, and paroxetine. So these are the selective serotonin reuptake inhibitors. We have the monoamine oxidase inhibitor or MyoIs, the transcypramine, phenazine, and moclobamine. Okay, then try the TCS or tetracyclines also are there, like imipramine, doxepin, desipramine, amoxicillin, trimixprim, and these are the examples of the tricyclics and the tetracyclics. Among this, serotonin reuptake inhibitor or the SSRIs are the most commonly used. Myois or myo inhibitors are also used, but these give rise to several toxicities and several side reactions. That is why these are the last resort of the antidepressants which are used. Okay. So these are all about the antidepressant drugs. So now I will go to directly to a topic which is very important in our medicinal chemistry course, and I have already discussed it for a couple of times, but this remains still the most important class of therapeutics of our, which we have discussed earlier. So that is the anti-cancer drug discovery. So cancer is resulted from a different reasons, and I have discussed it. You will find it in our various weeks also. We have discussed about the anti-cancer drugs, like we have discussed about the mm, uh, all this aspects, how cancer is generated from the defects in the cell cycle, and we have discussed the different inhibitor different classes of the anti-cancer drug. I will just summarize it today in little bit, not I will go to the details, but all the details you will find in our lecture material. Okay, in the week wise you will check the weekly materials, and you can just, you will find in all the slides, and in the previous slides which I have mentioned, you will find the details of the anti-cancer drugs. Okay, but today I will just go, I will just summarize a few topics of the anti-cancer drug discovery. So cancer cells have the defects in the normal regulatory controls governing the cell growth and division. Such defects arise from the mutation resulting in the activation of oncogenes and the inactivation of tumor suppressor genes. Defects in the signaling pathways are commonly found in the cancer cell. The pathways stimulating the cell growth and division are overactive as a result of the overproduction of a crucial protein in the pathway or the production of an abnormal protein. The proteins involved include the growth factors, receptors, signal proteins, and kinases. The production of the regulatory proteins which suppresses the cell growth and division is suppressed in many cancers. The cell cycle consists of the four phases. We have already discussed it, right? The four phases are the G1 phase, then the S phase, then the G2 phase, and then the M phase. Progression through the cell cycle controlled by the cyclin and cyclin dependent kinases, moderated by the restraining proteins. Defects in the cell system has been detected in 90% of cancer. Apoptosis is a destructive process leading to the cell death. Cells have monitoring systems which check the general health of the cell and that trigger the process of the apoptosis if there are too many defects. Regulatory proteins have a moderating influence on the apoptosis. Defects in the apoptosis increase the chances of the defective cells developing into the cancerous cells and reduce the effectiveness of the several drugs. Telomeres act on the species to stabilize the end of the DNA. Normally, they decrease the size of the each replication until they are too short to be effective, resulting in the cell death. Cancer cells activate an expression of an enzyme called telomerase, maintaining the telomerase becoming important. Angiogenesis is a process by which the tumors stimulate the growth of the new blood vessels to provide the nutrients required for the continued growth. Agents which inhibit the angiogenesis are useful in the anti-cancer therapy to inhibit the tumor growth and to enhance the effectiveness of the drugs. Metastasis is the process by which the cancer cells break free in the primary tumor, enter the blood supply, and set up the secondary tumors in the tissues. To do this, the regulatory controls which fix cells to a specific environment and which destroy the cells become detached and overruled. Surgery, radiotherapy, chemotherapy are used to treat the cancer. Chemotherapy usually involves combination of the drugs having different targets and mechanisms of action. Traditional anti-cancer drugs are generally cytotoxic. More modern drugs are selective in their action. Cancer cells can have intrinsic and acquired resistance to the anti-cancer drug. Resistance may be due to the poor uptake of the drug, increased production and the target protein, mutation which prevent the drug binding to the target, and alternative metabolic pathway or efflux system which expel the drugs from the cell. Intercalating drugs contain the planar aromatic or heteroaromatic system which can slide between the base pairs of the DNA double helix. Alkylating agents contain the electrophilic group that react with the nucleophilic centers of the DNA. If two electrophilic groups are present, interstrand or interstrand cross-linking of the DNA is possible. Nitrogen musters react with the guanine groups of the DNA to produce the cross-linking. The reactivity of the agents can be lowered by attaching the electron withdrawing group to the nitrogen to increase selectivity against DNA over proteins. Incorporation of the important biosynthetic building blocks aid the uptake of the rapidly dividing cells. Cisplatin is the analogous of the methylating agents which can cause interstrand cross-linking. They are commonly used for the treatment of the testicular and ovarian cancers. CC1065 analogs are highly potent alkylating agents which are being considered for use in antibody drug conjugates. Cholecystramine is a natural product which reacts with the nucleophiles to produce the identical species. Reaction with the DNA ultimately leads to cutting to the DNA chain. Antisense molecules have been designed to inhibit the mRNA molecule that code for the protein which suppresses the apoptosis. So all these things, that this is the brief summary of all these topics, but all these things I have already discussed in a very detail in our previous classes, that so you can just go through the material which is given to you earlier. And let us understand a few more key points regarding the anti-cancer drugs. So antimetabolites are the agents which inhibit the enzyme involved in the synthesis of DNA and its building blocks. Thymidylate synthesis catalyzes the synthesis of DTMP from the DUMP. The cofactor required for this reaction is regenerated by the enzyme dihydrofolate reductase. Inhibition of either enzyme is useful in anti-cancer therapy. Ribonucleotide reductase catalyzes the conversion of the ribonucleotide diphosphates and the deoxyribonucleotide diphosphates. It can be inhibited directly by the drugs or indirectly by the inhibiting adenosine deaminase. In the latter case, the buildup the DMATP results in the allosteric inhibition. Various nucleosides and purines act as prodrugs and converted to the cell to the agents that inhibit the DNA polymerases. The active agents also act as substrates and are incorporated into the growing DNA, leading to the chain termination or the inhibition of the replication. Hormone-based therapy is used against the cancer which are hormone dependent. Hormones can be administered which counteract the offending hormone. Alternatively, anti-hormonal compounds are administered to prevent the offending hormone from binding to the receptor. Glucocorticoids, estrogen, progestin, androgens, and LHRH are commonly used hormone therapies. Agents which act on the receptor antagonists are used to block the estrogen and androgen. Enzyme inhibitors are also used to block the synthesis of the hormones. An important target of the is the enzyme aromatase, which catalyzes the last step leading to the estrogens. Okay, already I have discussed about the aromatase inhibitors in our week 11. You can go through the slides of that particular group. Agents which inhibit the polymerization or depolymerization of the microtubules are important anti-cancer agents. The vinca alkaloid, podophylotoxin, combistatins, and a variety of other natural products bind to tubulin and inhibit the polymerization process. Paclitaxel and the derivatives bind to the tubulin and accelerate the polymerization by stabilizing the resulting microtubules. Newer analogs are being investigated which show better oral bioavailability, improved pharmacological properties, and activity against the drug-resistant cancers. A variety of natural products have been discovered which have the similar mechanism of action to the paclitaxel. Matrix metalloenzymes, the matrix metalloproteinases MMPs are zinc dependent enzyme which degrade the extracellular matrix and encourage the process of angiogenesis, tumor propagation, and metastasis. Inhibitor of the MMPs have been based on small peptide sequences of the protein substrate. In general, the susceptible peptide bond has been replaced by the stable bond. Substituent had been incorporated to fit the binding subsites, and the ligand for the zinc cofactor have been included. Second generation inhibitors of MMPs are non-nuclear, non-peptide in nature and are more sensitive in the action. The proteasome is destructive enzyme complex that breaks down the protein. Inhibition leads to the buildup of the completing regulatory proteins which triggers apoptosis. Histone acylases and deacylases are involved in the regulation of transcription. Inhibitors of the histone deacylases are in the clinical trials. A variety of other enzymes are potential target for the novel anti-cancer agent. Large number of synthetic structure natural products have anti-cancer properties with unknown mechanism. Others appear to work by having several different mechanisms. Protein therapy is also useful in the treatment of certain cancers. Clinical aspects of the antibodies and antibody drug conjugates. So antibodies, the trastuzumab is a humanized monoclonal antibody which targets the extracellular region of the HER2 growth factor receptor and was approved in 1998 for the standard first-line treatment of HER2 positive metastasis breast cancer in combination with the paclitaxel. HER2 is a member of the EGFR family of tyrosine kinase receptors which is overexpressed in 25% of breast cancers. When the antibody binds to the receptor, it includes the immune response to attack the specified cell. It is also promote the internalization and degradation of the receptor. Trastuzumab is given by injection. Unfortunately, the drug cannot cross the blood-brain barrier and is ineffective against any tumor cell and have metastasized in the brain. The drug resistance and cardiac toxicity are further problems which can arise. Alemtuzumab is a humanized antibody that lyses the beta lymphocytes and used for the beta cell chronic lymphocytic leukemia. Where other therapies have not worked, the antibody binds to the receptor, the CD552 antigen, which is found both on the normal and cancerous immune cells, the beta and B and T lymphocytes. Although the agent shows no selectivity for the cancer cells over the normal cells recover quicker after treatment. Rituximab is a chimeric antibody targeting the CD20 receptor on the beta lymphocyte and was approved in 1997 for the treatment of the diffuse B cell and non-Hodgkin's lymphoma and follicular lymphoma. It causes the lysis of the beta lymphocytes. Patients should have monitored very closely as there are reported fatalities relating to the release of the cytokines. In 2010, it was approved by the FDA for the treatment of the chronic lymphocytic leukemia. Cetuximab is a chimeric monoclonal antibody that targets the extracellular domain of the EGF receptor and blocks the EGF from binding. It is used alongside the irinotecan for the treatment of the metastasis colorectal tumors and which suppresses the EPGFR, which are proved against resistance and the previous chemotherapy and include erythrosin. The antibody is also used alongside the radiotherapy for the treatment of locally advanced squamous cell cancer in the head and neck. Bevacizumab is a humanized monoclonal antibody that is given intravenously and disable growth factor VEGF required for the angiogenesis. It is used in the first line treatment of the metastasis colorectal cancer along with the fluorouracil and folinic acid. It is also used in the first line treatment for the metastatic breast cancer along with the paclitaxel. Ofatumumab targets different epitope of CD20 receptor from the rituximab and was approved in October 2009. It is used to treat leukemia that cannot be controlled by other forms of chemotherapy. Panitumumab is fully humanized monoclonal antibody that targets the epidermal growth factor and has been approved for the treatment of the colorectal cancer. Antibody drug conjugates, the gemtuzumab or ozogamicin was approved for the treatment in the acute myeloid leukemia and was withdrawn in the 2010. Idimovseb was the first approved drug involving radioimmunotherapy for the treatment of non-Hodgkin's lymphoma. Tocilizumab was approved in 2003 for the treatment of the non-Hodgkin lymphoma, which was refractory to the rituximab, and this Brentuximab vedotin was approved in 2011 for the treatment of Hodgkin lymphoma and systematic anaplastic large cell lymphoma. The antibody targets cell membrane called CD30 and is linked to the anti-tumor agent monomethyl statin E, and which is a systematic analog for the naturally occurring orphan. So this was all about the anti-cancer agents. I have discussed in brief because in detail I have already discussed in our previous classes. So you can go through that particular slides and then you can read from there. Okay, and the last topic which I am going to summarize today is the cholinergic system. So the cholinergic signaling system. So what happens in the synapses involving the acetylcholine as a neurotransmitter? As you can see, it shows the synapse between the two neurons and even involved in when the message is transmitted from one neuron to other. Okay, the first stage involves the biosynthesis of acetylcholine. So biosynthesis of acetylcholine happens when the acetylcholine is synthesized from the coenzyme A and the choline with the help of the choline acyl transferase, and at the end of the presynaptic neuron, the reaction is catalyzed by the choline acyl transferase, and this is the structure of acetylcholine. Acetylcholine is incorporated into the membrane bound vesicle by the means of the specific transport protein. The arrival of the nerve signal leads to the opening of the calcium ion channel and increase the intracellular calcium concentration. This induces the vesicles to fuse with the cell membrane to release the transmitter to the synaptic gap. The acetylcholine crosses the synaptic gap and binds to the cholinergic receptor, resulting in the stimulation of the second neuron. Acetylcholine moves to the enzyme called as acetylcholine esterase, which is situated in the postsynaptic neuron, which is catalyzes the hydrolysis and the acetylcholine to produce the choline and acetic acid. Choline is taken up by the presynaptic neuron by the transport protein to continue the cycle. So what are the key points? The cholinergic nervous system involves the nerve which use the neurotransmitter acetylcholine as a chemical messenger. This include the motor nerves which innervate the skeletal muscle, nerves which synapse and other nerves in the peripheral nervous system, then the parasympathetic nerves in the cardiac and smooth muscle. There are two types of cholinergic receptor: muscarinic receptor, which is present in the smooth and cardiac muscle, and nicotinic receptor, which is present in the skeletal muscles in the synapses between the neurons. Acetylcholine is hydrolyzed by the enzyme acetylcholine esterase. When departs from the cholinergic receptor, the hydrolytic product choline is taken up by the presynaptic neuron and the acetylate back in the acetylcholine. The cholinergic receptor and the enzyme acetylcholine esterase are useful drug targets. Acetylcholine cannot be used as a drug because it rapidly hydrolyzed by the acid and enzyme. It shows no selectivity for the different types or subtypes of the cholinergic receptors. And these are the receptors classification. One is the ligand-gated ion channel and G-protein coupled receptor. Nicotinic acetylcholine receptors are the ligand-gated ones, and the G-protein coupled receptor is the muscarinic receptors, which are 5 type: M1, M2, M3, M4, M5. Nicotinic receptor are 2 type. One is the central nervous system plus automatic ganglia in the peripheral nervous system is a neuronal nicotinic acetylcholine receptors, and skeletal, which is the muscle the neural muscular nicotinic acetylcholine receptors, and these neuronal one was the homopentameric or heteropentameric. So these are the classifications of the different acetylcholine system. Okay. So this was up today. This was up to our summary lectures. So this presentation is uploaded to your particular course material, this summary session. You can go through that portion, and if you have any doubt or any query, you can drop an email to me. I will share my email ID. Okay, so if you have any doubt, any query, you can just let me know through the email. Before examination, I will suggest you any books or any explanation through email also if you have any doubt. Okay, so I will close the session for today. And thank you for being with me in this course of medicinal chemistry, and I am the closing the session for today. If you have any query, please let me know through this email ID.